Hi everybody. So it's a huge storm here today. Pretty stormy. Just a couple hours ago the road was completely impassable before the snowplows came. There was like a 4-foot drift. It's blowing so hard right now. So, but I still get outside no matter what. And I've lived in different climates, but this day to day is pretty stormy. I'm not going to go too far. Here's one of my ice chamois. So this is Lake Michigan. This is some of my property here, and it's a total whiteout pretty much. I mean, normally where I walk, you can't even see anything. So I'll just film for a little bit. It'll probably get really hard to hear me when I get out on the lake. But it's still good for you, creating a hormetic response or hormesis, which is a small amount of good stress for your body. So I'm just wearing my tennis shoes with no socks. And Juno doesn't have her coat on today. So usually you can see way out here. I'd say visibility, well, it's still not too bad. It's probably a half mile. I've seen it worse. But it's a pretty chilly one out here today. Imagine if you were one of the indigenous tribes from this area living here in a longhouse. This property that I have here was a village, native village for the Ojibwe. So Juno's ears are back. I think she's a little cold today. But it's still a beautiful day. Nature's full of vigor. Push yourself. Yeah, snowshoe weather before too long. Definitely off the ice. The woods is definitely snowshoe weather, but it's been pretty good on the ice up until today. But anyway, I just wanted to share with you, the importance of getting outdoors in every climate every day. Just for a few minutes. A lot of people commute to work and they walk out to their 2-car attached garage, get in their car, drive to work on the freeway for an hour and a half, maybe stopping to drive up, get some coffee somewhere along the way. And then some people pull into their parking garage and get in the elevator and go up to their office. And then at lunchtime, they take the elevator down to the subterranean cafeteria. And that's a little open water there. There's a little creek that comes out. It's only about a foot deep or so. And then they, it's a little slushy. They come on back home after work, get in their elevator, take the elevator down to the parking garage or up to where their level is, and then they drive on home, pull up to their mailbox, roll their window down, and have their outdoors for the day while they're getting their mail or the paper. And then they pull back in their 2-car garage, make dinner, or pick it up on the way home, and eat dinner, read the paper, open the mail, watch TV, go to bed, get up and do it all over again. And we wonder why we're fat, sick, and nearly dead and can't remember anything. We're not getting outdoors. We're not creating any hormesis for our body. So live like your ancestor. Live like your great-great-grandfather and great-great-grandmother. Were they shoveling snow? Were they driving a Model A or a Model T on dirt roads and shoveling their way out and What kind of exercises were they encountering? What kind of weather were they encountering? What kind of food were they eating? Where were they from? So live like your ancestors. And if you do that, even if it's an artificial recreation of living like your ancestors, you'll be a lot healthier, happier, and you'll have a greater health span and live a life full of vitality with longer longevity, maybe than your parents. So the power is in your mind and in your hands and in your heart. So get outdoors and go wild and follow your wildest dreams. Why won't that let me do that? I'm gonna pee.
There's certain gene polymorphisms. I know there's one in the PPAR alpha gene, which is important for the whole process of making ketone bodies, and that people have that, they actually can't do that very well, and so it can be sort of dangerous, and they can have inflammation. Sort of the opposite profile will happen, so it's certainly something to consider. And then, not to mention, like you found in your paper that you just published this year, was it last month?
Yeah,
Last
September,
month, that you found, there were multiple changes going on. There was there was a decrease in insulin, obviously,
Yes.
in IGF-1, and mTOR activity
Yes.
went down.
Yes.
And then So that's something you're not going to just get from beta hydroxybutyrate. Maybe, we don't know.
We don't know. I suspect not for the mTOR part, but we also saw, we compared the ketogenic diet to a high-fat diet, which was not ketogenic. That is, in which mice ate a large amount of fat, but had enough carbohydrates to suppress ketogenesis. And one of the biggest differences, I think, actually, between these two was the activation of PPAR alpha. And this is also very exciting because that indicates that maybe a lot of the beneficial effect that we see on the ketogenic diet come from PPAR alpha activation. And there's, we've been reading that literature. There's quite a bit of information there that really hasn't been pursued as a next direction to try to dissect these effects.
Because PPAR alpha is doing something to the mitochondria, or what's the main?
It's a key enzyme of the fasting response, but it seems to be more highly activated in response to the ketogenic diet than to a pure high-fat diet.
What about fasting? If you compare fasting to
it is activated. We did not compare to fasting. We compared it
just
high fat to, but I think it just points to a new direction in which we can start dissecting what is the role of PPAR alpha in these responses.
But with your diet, you limited the protein intake and you didn't limit the caloric intake. That was the other study?
No, the protein intake was actually 10% isocaloric between all of the diets that we tested, and for the other group did the same thing. So we were very careful not to change the protein intake. In the other group, actually, they did change a bit the protein content, which could be taken as a confounding variable.
Right,
so I think we were very careful in not changing the protein content.
Is that standard for ketogenic diets have grown in popularity, and so do you think that if the ketogenic diet you used, you hadn't have done a 10% protein, if they had done a little bit more? I'm not sure if maybe that technically wouldn't even be a ketogenic diet, but would it have changed the IGF-1 mTOR axis as much?
You're bringing an important point, which is that a number of people who go on a ketogenic diet tend to compensate by increasing their protein intake, which might actually put them at risk from exactly what you're describing, increased IGF-1 signaling and increased actually risk of cancer. There's a really close correlation between your IGF-1 level and your risk of cancer. So I think this is something that needs to be considered in the future for people who are on a long-term ketogenic diet.
And with the caloric restriction diets, I think I read, there was a study I remember reading that humans that end up doing caloric restriction, like there's the whole society of caloric restriction.
Yes.
They end up eating they eat 30% less calories than they normally would, or whatever, something like that. But they end up eating a higher percentage of protein because it's more satiating. So humans sort of naturally gravitate to eating more protein when they're eating less food.
Yes.
And so what's interesting is a lot of those, I think you even published a study recently looking at biomarkers of aging in lymphocytes or monocytes.
Yes, and we saw no difference between people on calorie restriction. This points again to, and this is something important to discuss, to consider when a lot of the work that we do in the lab are done on mice that are isogenic or congenic. So these are mice that are all similar to one another. When Steak takes discoveries and when you bring discoveries to the human population, it is critical to take into consideration the incredible variation between different people and how they might respond to the same interventions. We know from everything that we've studied in medicine that I am not the same as you are, and you are different from your neighbor in many biological responses, and that includes response to calorie restriction. I suspect that in a number of people it would actually do them harm, and in a number of people it will do them good. We know this when we test different strains of mice by calorie restriction. Half of the strains actually responded by a shortening of lifespan, the other half by an increasing lifespan. So we take calorie restriction, for example, as a universal modification that will increase lifespan. It's not what is seen in the literature. And I would say the same is going to be even more true for humans. So I think this brings for me brings up one of the key things that is lacking in the field of aging is the identification of biomarkers that will allow us to test on an individual basis whether the intervention or the modifications that you're imposing is actually pointing you in the right direction or in the wrong direction. And I would caution anyone who's considering doing something long-term in terms of their health to be very careful in how they feel and because we don't have these biomarkers of aging. They're emerging, but they're not validated. It's certainly not in the human populations. They're emerging in mice models and other models. But that's really where we will be going in the next few years, is really having true biomarkers that allow us to predict whether a given intervention is beneficial or actually hurtful.
Yeah. What do you think are the top three right now we have for biomarkers for aging?
Well, there's I can tell you the one that are really exciting me. There's Alexander Zhoronkov as a company called In Silico Medicine that is building biomarkers of aging based on facial recognition and based on metabolites. Just consider how
metabolites.
So consider how you and I can look at a human and pretty much guess what their age is for most people, and consider the fact that where this is coming from is the fact that we've met thousands of people and we've heard about their ages, and we sort of build a database of information. So what Alex and his company have done is they've taken the pictures of thousands of people and fed them to a deep neural network and artificial intelligence, and the computer has been able to learn how to recognize and to link on average, what is the face of a 70-year-old versus a 60-year-old? So you can do this, and it will tell you what the computer thinks your age is. And for some people, you fall right on, but for a number of people, you find that your age based on your face actually looks younger or older. So this would be a reflection of your biological age. He's also been able to do this with blood markers, with 20 or 30 or 40 usual blood markers. By screening a large number of people, he can actually generate standard curves, and then you can put in your blood values and see where you fit. So that's one approach. BioAge is a company in the Bay Area that is actually pursuing the same idea, looking at artificial intelligence and biomarkers in plasma or in urine or and so on for identifying your age. And finally, one big area is the whole idea of the epigenetic clock.
Yeah, Steve
Horvath and Trey Heidecker have shown that there are changes in DNA methylation that actually predict pretty closely your chronological age.
It's pretty cool.
So I think all of those together are emerging as indicative that there are markers that we can reliably measure. The key is, how many of those do we need? What are how can we reduce this to something that's going to be very strongly predictor, not so much of your chronological age, but of your biological age?
Right, yeah. I think there was a study I did read, maybe it was the company that you mentioned, where there was a panel of blood biomarkers that they looked at, like telomere length and immunosenescence and, the standard panel. And then they had people, I think it was people that were asked to identify their facial age or something like that. And then they asked their chronological age, and their facial age actually matched their biological age more than their chronological or something like that.
Exactly. We all know in our acquaintances some people that actually look younger and some people that look older. And we know this because we have a deep neural network in our head that has allowed us to build, a reference point so that we can pretty much guess how old people are. And the computer does it even more efficiently because it can be fed tens or hundreds of thousands of pictures.
That's cool. I want to try it out.
Yeah, it is actually really cool.
I just sent off my, I'm going to be getting my telomeres tested. I don't know how reliable that's going to be. We'll see. But, I've been getting less sleep than usual because I'm a new mother, and that's been shown to affect. But it'll be interesting to see. Anyways, we digressed. I kind of wanted to ask you a little bit about, we're talking about fasting. We talked a lot about, insulin signaling decreasing. IGF-1, we've talked about before in the podcast with Walter Longo and others, just how the protein restriction seems to really regulate that, and that also regulates the aging process, mTOR as well. But something else that really seems to change with fasting is the NAD levels.
Yes.
And that's something that your lab has studied extensively with the sirtuins, and so maybe could you talk a little bit about, because NAD is also extremely exciting to me, and it's pretty popular these days as well.
Yes. So NAD has emerged as one of these critical intermediary metabolites. Think of ketone bodies, NAD are all, I call them currencies. I mean, so think about your organism as a country. You need to circulate energy, and NAD is one way that our body is utilizing within the cell to transfer energy. It's almost like the Brinkman truck that carries the money. And NAD can actually is a so-called hydride acceptor. While we oxidize food, it can actually serve as an acceptor for electron, and it can transfer them, for example, to the respiratory chain. So it's one way for the energy to be circulated within the cell. And there's growing evidence that its level decrease during aging. Why that happens is still one of the big mysteries. And so this has yielded a whole approach that trying to understand first, what are the consequences of decreased NAD levels? And one of the consequences is that enzymes like sirtuins, which rely on NAD to exert all of their beneficial activities, actually function less well. That's what happens during aging. But also many other enzymes that are involved in our metabolism are relying on NAD, and so they function less well. So your intermediary metabolism functions less well. The sirtuins, which are global regulators, function less well. Anyway, so I lost.
You're basically falling apart.
Yeah, essentially, everything becomes a little less efficient. So out of these discoveries came the idea that maybe we should replenish the decreasing level of NAD, and so this has yielded some discoveries such as nicotinamide riboside, nicotinamide mononucleotide, which are now being taken by a lot of people, and with the hope that it will, correct some of these problems. One word of caution, I think, there is we do not know why these levels decrease. They could decrease because we have decreased production of NAD, but it could also decrease because we have accelerated destruction of NAD, which means if it's accelerated destruction, bringing more into it is sort of like pouring more NAD in the leaky sink. So I think a lot of our work right now is trying to understand what is the cause of the decrease in NAD during aging, because I think it will yield very different solutions. If you find that there's a leaky sink, we'll work at plugging the sink versus keeping pouring water.
I have a theory.
Yes.
So I know, coming from, I did a lot of work with DNA damage.
Yes.
And knowing that one of the main enzymes.
PARP.
Exactly.
Yes.
I mean, if you think about, so one of the main enzymes that repairs damage as we age, DNA damage, PARP, requires NAD, and it's like, if you're accumulating more and more damage as you age, you have to repair more of that damage, and the more and more damage you're having, maybe it's sucking the NAD, sort of like almost a triage where it's like, well, you got to keep repairing that damage, so then other things like the mitochondria suffer, so. I
completely agree, and actually there are two major theories right now that have been proposed in terms of why does NAD go down. One is activated PARP, and indeed, as we age, we accumulate DNA damage. It's been shown, especially in the brain recently. And so the idea is by activating PARP, you can constantly deplete your NAD levels. The second one is we all have in our body a so-called salvage pathway for NAD, because NAD turns over. There's a so-called salvage pathway that allows it to be recycled back to, so we can get NAD from the food, but also we salvage the one that we utilize.
Right.
And this salvage pathway has been shown to becoming paralyzed while you age. There's an enzyme called an AMPT that has received a lot of attention. That enzyme tends to be inhibited by chronic inflammation. and a high-fat diet. So one way, so it could be a combination of both of these things, but it could be other mechanisms. Actually, we're working yet on another mechanism, which is that there might be accelerated destruction by other enzymes beyond PARP, and getting some exciting results in this direction.
Cool. So for people that are not familiar with why, like in terms of what role it plays, in the aging process, it seems as though, at least in some of the studies that I've seen, that mitochondrial function really seems to be important. And
Yes.
I know that you've shown that the sirtuin 3 in the mitochondria itself seems to be really important for the mitochondrial function, keeping your mitochondria young. And that mitochondria play a very important role in aging as well.
A critical role. Many of the aging pathways that we know, be it the unfolded protein response or mitochondrial biogenesis, all point to efficient mitochondria as one of the key ways to stay young. And one reason is because this is a way, so you can generate energy from glycolysis, which is glucose, carbohydrates, which is we know is linked to aging, but also via mitochondria via a process called oxidative phosphorylation. And that process is not necessarily depending on glucose, but is dependent on an efficient mitochondrial function. So we talked about NAD-dependent enzyme. Many of these enzymes reside in the mitochondria. And we found that, for example, the NAD supplementation that is being tested in a variety of aging model requires SIRT3 quite often. This mitochondrial sirtuin, yes.
Nicotinamide riboside does?
Yes. For example, the paper we've published was focused on the age-associated or noise-induced loss of hearing. So if you actually subject mice or humans to very acute hearing, acute noise, they have a dose-dependent loss of hearing. You can protect mice completely from this effect by supplementing with NAD.
Wow.
But with nicotinamide riboside. So if you're going to a rock concert and you want to protect yourself, and this is definitely something
Harley-Davidson riders.
Yes. In mice, it actually had an enormous effect, and we found that this effect was dependent on SIRT3. So in SIRT3 knockout mice, the effect was lost.
Do you know if if that was the SIRT3 in the mitochondria of stem cells, or was it just any cell, or do you not know?
In the case of hearing loss, it's dependent on some really uniquely sensitive cells in the inner ear, but generally there's been a sort of an assumption in the field that most of the T1, I think
Oh, really?
Yes, and in some cases it is. In this particular case, noise-induced hearing loss, it was really clearly SIRT3.
So even the studies that have looked at, for example, I think there's there was a mouse that had some sort of mitochondrial disorder.
Yes.
Even that one was dependent on SIRT1.
No, actually, I did not. In that case, I think the assumption was that it might be helping global mitochondrial function. And so there's a growing number of cases, in, for example, DNA damage-associated disease, where you see accelerated aging, where people have been testing the effect of supplementation, because those conditions are associated with increased PARP activity, just like you mentioned. And in those cases, I think it hasn't always been clearly mapped what is the real target of NAD that is dependent on for the beneficial effect.
Do you know if so NAD levels do decrease with age? Do you know, has it been looked at, like, for example, in animals, like in rodents, when they're fasted, because fasting increases NAD when they're fasting and they're older, does that help rejuvenate the NAD irrespective of their age? Does it?
I don't know. I don't know the answer to that question. It's a good question. I think, one thing that I remember also about fasting, another critical effect of fasting that is really essential in our thinking is autophagy.
Yes.
And so, there's growing evidence also that autophagy by itself responds to acetyl-CoA levels. This is a new area that people are starting to work in, and including my labs, trying to understand what is it about acetyl-CoA, which is another, think about the currency exchanger in the cells, intermediary metabolite, that appears to have not only role as a nutrient, but also as a signaling molecule. Acetyl-CoA is in the acetyltransferases, which are the opposite of what we were talking about the HDACs earlier. So
Yeah,
I think there's really a lot of crosstalk between all of these pathways, and we're working very actively now on modulation of signaling via acetyl